I'd like to thank the organizers for inviting me and all of you for being here. So we're talking about the next generation sequencing-based detection of MRD in ALL, and then I'll have a couple other slides and other lymphoid neoplasm also. So what are the issues in MRD testing? What is it that you would like to see in a test that is for MRD testing? So you want it to be reliable, obviously, and reproducible. You want to be able to comprehensive of patients, so you want to be able to assess as many patients as possible. You want to be able to look at the clonal heterogeneity and evolution as the cancer, which, as we all know, does evolve. You want it to be sensitive, and at the same time, you want it to be a turnaround time that will be reasonable from a clinical sense. So our platform is based on the marine receptor sequencing. I won't go into too much detail. I assume all of you know about the VDJ recombination that produces the immunoglobulin heavy chain variable region. That is basically every B cell at the end in its development is going to create a specific VDJ sequence that is on its DNA that is unique to that cell and its progeny. So it's basically a barcode for that cell and its progeny that can be assessed by the sequence. The premise of the technology is that we can have a sample and amplify all the VDJ sequences in that sample and be able to basically have their full repertoire, all the clones that are present in that sample enumerated, and you know how many molecules they present in that blood sample or bone marrow sample and other sample. So in the lab, basically the workflow for the assay is that you get blood and bone marrow from the patient that gets shipped to the lab where DNA is extracted. Then amplification, multiplex amplification is performed where all the V and J sequences are amplified. So whatever is the rearrangement that is present in that repertoire will be amplified by at least one of the pair of primers. Then there's a second stage amplification where sequences that are important for the sequencing reaction to occur are appended. And finally, you go in the sequencing machine, produces a lot of reads that get analyzed. And the result of all that is what we call the primary analysis, which is this clone or that sequence, you have of it 2, that one you have 5, and so on and so forth. So you get this full repertoire, and the question is, what do you do with it? How do you know what sequence you want to follow? And in the context of leukemia or lymphoid neoplasm in general, you identify it by looking at the diagnostic sample, because the diagnostic sample is loaded with the cancer cell, and hence all the cancer cells are all clonal, coming from one original B cell or T cell, and hence they all have the same barcode. So you get that leukemic bone marrow sample diagnosis, and we perform the different assays that we have, which is immunoglobulin heavy chain, the kappa chain, the TCR beta delta gamma, and then this is the cartoon of each sequence is one dot, and basically you have the log 10 frequencies on the y-axis, and you identify the 2 clones that are specific to the leukemia in this patient. So you have to do this for each patient. Each patient got their own sequence that is specific for that patient. So you identify for that patient that they have an immunoglobulin heavy chain sequence that's known, that's the sequence of that clone, another one, the TCR delta, which is the sequence of that clone. So once you know that that is the sequence for those patients, and this is the receptors that are worthy or informative and hence should be followed in the follow-up samples, when you get a follow-up samples, you would only do the amplification of those 2 receptors. You're amplifying the full repertoire but for those 2 receptors, but you're only amplifying those 2 receptors, and then you can identify the clone at now 1 in a million or below. Great. So just now mention on the various criteria that I mentioned initially as to what's relevant for MRD testing. So first, reliability and reproducibility. So there's a lot, obviously, of effort to go from publishing a paper to actually get a test that's clinical grade testing that we can provide clinical services as well as clinical trial services. And you need to, it's kind of going from the 80% works to 99.9% works. And basically for that to happen, a whole bunch of different optimization needed to happen, from being able to work with different sample types. So you can, like slides, so you can identify what the clone from archived sample, to being able to work with, make sure that you get reliable results, whether you have fresh samples or older samples. A fresh bone marrow, that's 1 day old or 4 day old or 1 week old. How do you manage to do sample QC in a way that allows you to measure effects of heparin and so on that's known to inhibit PCR. PCR amplification quantitation, how do you make sure you have standards so that you can basically, the quantitation that you have for all your, you don't have any basic amplification bias and all the different products that you generate. How do you go from relative to absolute quantitation? How do you identify the cancer clone? And finally, how do you detect evolution? So there's lots of kind of technical details that I won't go into each one separately, but I will focus on, after doing all that, you're able to get very nice validation and lots of clinical studies. So we've now published about 10 MRD data, MRD papers in different lymphoid neoplasm, validating the utility of our technology. This is, an old slide that hasn't yet been updated, except I updated for this presentation, the ALL column of it. The point of this is to say that we've done lots of studies in various diseases. As I said, it's probably a year old now. The total number of samples that we've done is probably now over 10,000 sample in the research setting. I'm not counting the clinical service setting. So these are all samples that have been done where we have assessment, clinical validation by assessment of comparison with flow cytometry, allele-specific PCR, or outcome. So this is the first paper that we published with Dr. Campana when he was at St. Jude, where we compared flow cytometry and sequencing. So what we get here is flow cytometry and sequencing. When they're both positive, we get a very nice correlation, where the correlation is essentially 1-to-1. One is used. We see all this wealth of information about 0.01% being predictive, and here, if you do it by sequencing, you don't need now to translate what this 0.01% by flow means by sequencing. It's the same number. If it's 0.01 there, it's 0.01 here. There's nice correlation of positive. There's a lot of negatives in both, but there is now a set of samples that are positive by sequencing but negative by flow, because they are below the level of sensitivity of flow cytometry, which is 10 to the minus 4 or 0.01%. This is a comparison now with allele-specific PCR with Dr. Wendy Stock data, where again we have a very nice correlation between the two platforms. Again, we get this sensitivity advantage with the sequencing, and then there are some clones here, some samples here that are positive by ASO and negative by sequencing. Obviously, we don't know what happens to them. It is, there is 8 points here, and 3 of them have other receptors tested by ASO and by sequencing, and they all look negative. This is yet another validation with the Euro-MRD. Euro-MRD have been doing allele-specific PCR for years, and so this is comparing with, and this is actually a slide from Dr. Bruggeman's presentation at ASH, so I just copied it exactly, so it's a different format. I apologize for that than the rest. But basically, this is the results from allele-specific PCR compared to by sequencing. Again, there's a very nice correlation. There is this gray zone of what they call an allele-specific PCR is below quantitative level, so it's low level by allele-specific PCR, but it's below their quantitation level, and you get a bunch of positive. And again, you get some positives, really, advantage of sensitivity by sequencing compared to PCR, but then there's a whole lot of negative in both techniques. So a lot of hopefully that convinced you, there's a lot of technical validation compared to the standards that are out there, flow cytometry and sequencing. So beyond reliability now, the question is the rate of identification of malignant clone. How often are we able to detect the cancer clone in those ALL patients? And let's skip that. So we have a set of studies of about, I think here we have 11 that we've now completed. 11 studies that we've done with various collaborators, different patient cohort that add up to 579 patients, where we have diagnostic sample and ask how often are we able to identify a cancer clone. And the answer is the vast majority of cases we are able to do that. So generally, basically, we will be able to find a cancer clone, more than 95% of the cases of ALL. Tracking multiple clones, or So when we identify a clone, so in that graph is the question, do we identify a clone, do we not identify a clone? When we identify, we may identify more than one sequence, not just one. And just to say, there's here a little bit of confusion when I say clone. When my wording says clone, it doesn't necessarily mean it's a different cell. It could be two different sequences in the same cell. I'm not making any inference about whether they're two different cells or one. So, but the question is how many sequences do we get to follow? And you see that, there's the mode is at 3, very infrequently we get 1, mostly we get 2 and 3 and so on and so forth. And the advantage of that is that obviously when we have more than one sequence, you can follow more than one sequence. Now I'll show that in a little bit. So that's one dimension of how many clones do we see at the time of diagnosis that you have to follow. But in addition, this is work that we've done with our Stanford collaborators, Dr. Gawad, Dahl, and Lacayo, where we looked at evolution of the cancer clone at the time of diagnosis. So at the time of diagnosis, we find a cancer clone, which is defined by its high frequency and looks like a normal, like any other immunoglobulin heavy chain with the V, D, and J and N bases in the middle. So that is a normal-looking immunoglobulin heavy chain. But what we found is that there is a lot of other related sequences that have the same J, the same D, some of the same ends here, and some of the same ends here, but not the full. And what that is basically, that's basically what happens is you get a V replacement where the initial clone, the V segment of that sequence get replaced by another. And that's an old phenomenon that has been described in ALL literature, the V replacement. But what we have shown is here is the extent of it is, since we can look at those cells at very low frequency, we identify a lot more evolution than has been discovered before. And we've seen that 50% of ALL patients have evolution at the time of diagnosis. This is an example where case where there was more than one clone, where clone was evolved from another one. So we found 3 sequences at the beginning. And one of them with the treatment, this is in collaboration with Dr. Stock. With the treatment, one of those 2 clones go down in frequency, while the other 2 don't. Now, Dr. Stock had made allele-specific PCR probes, as it turned out, for that clone. So it seemed like the patient is responding, but in fact, the patient relapsed a month later. It's obviously, now that you see those 2 clones here that have not responded to treatment, it's not surprising that's happening. This is again from Dr. Brueggemann, our German collaborator, slide from ASH last year, and this is kind of a complicated slide, different patients. This is where we had diagnostic and relapse samples. And typically, what we should focus on is this is a log 10 scale, so everything is, you really want to focus at the very high frequency things, which is basically here. So often the diagnostic and relapse clones are basically the same, 1 or 2 or so on, they're all the same. But sometimes it's not. So that the relapse is not a diagnostic clone. It's a new clone that have evolved. And in this case, even though it's a new clone, we could tell without having seen it before that it has evolved from the diagnostic clone, because we could see that it's phylogenetically related and the same mechanism of V replacement that we described. So even though we didn't see that clone at the time of diagnosis, we could still predict that we could still call MRD in that sample and say, yes, there is MRD, because that is a clone that is the result of the evolution of the diagnostic clone. Sensitivity is the next set of next criteria, and in this paper we had shown the technical validation of sensitivity where we diluted. This is with Dr. Campana's paper that was published a couple years ago, where we diluted 12 tumors, 12 leukemia into healthy bloods in duplicates and looked at their input, expected frequency, and observed frequency, and seeing that basically there's a very nice correlation, repeatability, and accuracy. Obviously, this is being able to predict prognosis. This is a result from unpublished results with our Dana-Farber collaboration. And this is in childhood leukemia. So, it's not surprised that we can predict prognosis at the 10 to the minus 4 threshold, which is basically, since we have a very nice correlation with flow, it's not surprising that we're able to duplicate what flow has shown, and Dr. Hunger has just eloquently showed us. But we do see that, in addition to those, if we just focus on the group that have a low-level MRD, so just on the 10⁻⁴ to the -6, so with low-level MRD, they have a worse prognosis than those guys that are negative. Obviously, it's not as dramatically, the effect size is not as dramatic as if you look at, obviously, 10⁻⁴. So obviously, the effect size is smaller, but there is an effect nonetheless. And obviously, that has to do with what time you are actually testing MRD. At what time point during treatment would you test it? So turnaround time, so I mentioned that, so we have a CLIA-certified CAP-accredited laboratory that provides clinical service in the US for physicians sending us basically the blood sample, and we send back a report within a 1-week turnaround time. Basically, the report, all the reports sends back, positive, the level, and the serial sampling, and delivered through a secure online portal. I will end with discussing validation in other lymphoid neoplasm, and I will start with another leukemia, chronic lymphocytic leukemia. This is a collaboration with Dr. Logan, who will be presenting on other collaboration we have together in ALL. So this is this collaboration focused on post-allo-transplant CLL patients, and this is a landmark analysis 1 year after transplant. So at that time point, the graft versus leukemia effect has effectively taken effect over the first year, and at the 1 year, you can basically ask, does the patient still have MRD at that time point or not? And patients who still are MRD positive at 1 year do poorly, versus those that are free of disease at 1 year end up doing really well. So in terms of looking at, in this analysis, we didn't just look at the landmark analysis, but we had actually serial samples of those patients post-transplant. So when we now took at a couple example patients from the two graphs, the MRD negative and MRD positive, the blue patient you see goes down in frequency, the blue to, this is days, so at 9 months become free of disease, and they still are disease-free 6 years later. On the other hand, the orange goes down in frequency to pretty low level. This is now 30 per million, or basically less than 1 in 10. This will be the 1 in 10,000. So this is 1 in 30,000 essentially. But it doesn't come, so it's pretty low level, but it's still not zero. And at that point, then the patient's level starts increasing until the patient relapses a little after here. Going to other diseases and multiple myeloma, this is a collaboration we did with our Spanish collaborators, Spanish consortium, where we looked at, again, landmark analysis at end of treatment in this cohort where we have MRD negative patients and MRD positive patients. So this is the overall survival, and this is time to progression. You see that there is a stark difference. So this is 4 years down, and you're able to have a pretty big difference in the prognosis between the MRD negatives and MRD positives. This includes patients who are in CR, in complete remission, as well as VGPR, very good partial response. So the question is, if you take out the VGPR, if you just look at CR, those are patients that we, in a clinical setting, right now, that's the standard that we can't distinguish between all of them. They're all supposed to have the same prognosis. But now, if you take those CR patients and stratify them based on whether they are MRD negative or positive, you get, again, a stark difference in the prognosis over years, let's say 4 years after taking the test. You have patients who are surviving up to 90% versus those below 40%. Another application is in the lymphomas, and I will restrict myself to diffuse large B-cell lymphoma. So this is in collaboration with the NCI, Dr. Wyndham Wilson. So this is basically looking at patients who have finished the course of treatment, the induction and treatment, and now they are in surveillance. So they are in CR, and they get CT scans in a regular fashion, and each circle is the time that they get the MRD test, but every time they get an MRD test, they also have a CT scan. So all those white circles are also accompanied by a CT scan that's that's negative. And then the square is when the patient relapsed based on clinical symptoms and CT scan being positive. So you see that, in some patients, like patient 48, the patient relapses and the MRD become positive or at the same time. But in other patients, that there is a lead time where the patient become MRD positive before they relapse. What's exciting about this is the really low level of false positives. So we have tested, 90 patients, and 700-800 time points that were tested, and essentially there is when the patient has a positive result, essentially all the time they relapse, which is very compelling. So summary is that ClonoSIGHT is a validated, reliable platform for MRD detection in ALL and other lymphoid neoplasm, and has attractive feature, hopefully that may compare favorably with flow cytometry and ASO, including to be able to track multiple clones and evolution, the high identification rate of cancer, and sensitivity and turnaround time is equivalent to, or probably slower than flow because we take a week, but it's still probably clinically relevant. Thank you.